This morning we've got a really nice session on nosocomial infections and MRSA, and it's a pleasure to invite the first speaker to the podium, Ed Feil, who will be speaking on biology and epidemiology of MRSA from population genomic approaches. So I guess we just pull it up like so.
Great. Good morning, everybody. I'd like to start by thanking Mark and Bill and the organisers for inviting me here. It's been an absolutely fascinating meeting so far. So I'm going to be talking a little bit about some of the work I've been doing on all these next-gen sequencing data that are starting to flood forth from the gates of the Sanger Institute. And I've just been thinking about and working through just exactly what added value these might add to our understanding of the transmission dynamics and the microevolutionary processes in MRSA. So as I'm kicking off the session this morning on MRSA, it's just a very brief introductory slide about the organism. Low GC gram-positive firmicute. Similar to the pneumococcus, as Bill pointed out yesterday, a key characteristic and often forgotten characteristic in terms of its population is that it is essentially a commensal species rather than a pathogen, and cases of invasive disease are massively rare compared to frequency of asymptomatic carriage. If it does cause clinical problems, it causes a very wide range of symptoms, ranging from very mild boils and carbuncles to life-threatening endocarditis and other conditions. So it's been well publicized, and I'm sure you're all aware that the public health burden of Staph aureus has been massively increased due to the acquisition of beta-lactam resistance, the MRSA methicillin-resistant Staph aureus throughout Europe, Japan, and North America. Although in the UK, I'm happy to say that rates have dropped off approximately halved in the last 4 years or so. There was a general election in the UK in 2006, I think it was, when the hospital hygiene and the spread of the superbugs was one of the big agenda points, one of the big six of the campaign. And the government set these targets that MRSA rates would be halved by these hospital deep cleans and all the rest of it. And those of us in the business rolled our eyes and said, of course, yeah, okay, say what you like. And the bugs must have read the targets, because they hit them absolutely bang on. So it hasn't been widely reported in the UK, but everything seems to be moving in the right direction, at least, although, of course, it is still a major burden. So the spread of beta-lactam resistance is due to the horizontal dissemination of a large chromosomal cassette, the so-called SCC MEC element. This varies in size from 20 kilobases to over 60. There's various different types, and there's a whole very active field of research in the evolution of this particular element. And of course, as you're well aware here in the States, particularly, MRSA is an increasing problem in the community. You have the USA 300, which hasn't, it's arrived in the UK, but doesn't seem to be getting it's being slow in getting a significant foothold at the moment. So when I say I'm talking about Staph aureus, I'm actually talking about ST239, which is the lineage that I've highlighted on this tree. Apologies for the lack of annotation on this tree. This is actually data that's 1 week old, based on 500 genome-wide SNP data sets of the whole population. So it neatly shows where the ST239 lineage that I'm talking about. So the ST stands for the sequence type, as characterized by the MLST, which Bill nicely introduced yesterday. Is there a pointer anywhere? Okay, thanks. One of the curious things about the evolution of this particular lineage is that it's absolutely a beautiful example of this sleazy cut and paste sex, to borrow a phrase from yesterday. Actually, this still, to my knowledge, represents the largest homologous replacement known in bacteria. Something like 20% of the genome has come in from an unrelated lineage, ST36, has come into an ST8 background. The figure at the top shows SNPs in common, so it's TW20, which is the reference genome for ST239. Thank you. So where a SNP in, is it the top button? Ooh. I'm being dense. That one.
The other top button.
The other top, oh, I'm holding it upside down. I blinded myself. So where there's a SNP in common between the ST239 and the ST8, it's shown as a mark, or the ST36, but different in the other genome. So you can see this it passes through. This is just one event. It passes through the origin of replication. So it's by a completely unknown mechanism, but it's quite a striking observation. So more to the point, the relevance of ST239 is that globally, I would argue it's by far the most common hospital-acquired MRSA lineage. It's essentially disappeared from Western Europe. It was one of the early emerging MRSA lineages. But if we were to go through mainland Asia, or in fact all the way from Eastern Europe, all the way over through Russia and Central Asia, all the way to China, as far as we can tell, if you were to get an MRSA in a hospital in any of that region, it would be roughly 90% chance that it would be an ST239. It's also extremely common, predominant in South America. It's multiply resistant. It's the only lineage that has the so-called type 3 SCC MEC element, which is the really big one. And it's really only found within a hospital environment. It's there's only a couple of cases where it's actually been picked up in the community. So it really does look like it's a hospital-adapted lineage. There's epidemiological and experimental evidence that it's actually has increased invasive potential and biofilm forming capabilities, and the spread of it does indicate that it's highly transmissible between hospitals. So it came to prominence again in the UK. There was an outbreak in a London hospital in 2007, I think 2008 maybe. And after this outbreak, which actually spread beyond a single hospital, but this is after this outbreak that the reference strain TW20 was sequenced. So then we used this reference sequence to try out this new Selecta technology. This is work which was published early last year, Science. So the idea was to look to see how useful the utility of this sort of approach would be for looking at transmission patterns. And we decided to basically do two studies in one go, looking at the extremes of scale, both from a global perspective. So 42 isolates were recovered from a very wide geographical range. And we supplemented that strain collection with another 20 strains, of all of 239, this time from a single hospital. So we're really going from one extreme to the other. So we were looking for both the global spread and the potential for looking at local transmission dynamics within a single healthcare setting. So there were about 5,500 high-quality SNPs called, so not many in the 60 isolates. So we could clearly identify the well-characterized mobile genetic elements in the aureus genome, the phage transposons, the SCC MEC, of course. These MGEs corresponded to high SNP density, so in order to make any robust phylogenetic inferences, of course, we had to remove these so-called accessory elements. And we came up with a working definition of what we would call core and non-core, based on the fact that we'd only include regions of core if they were present, regions that are at least a kilobase long, which were actually present, that is mapped, in all of the 63 isolates that we looked at. So, of course, the accessory elements showed a lot higher SNP density and a lot lower coverage than the core. So this is what the SNPs look like. You can see the well-characterized MGEs down the bottom. There's regions of high SNP density which more or less correspond to the phage and the SCC MET. You can see there's a big phage here which is missing in most of the isolates, hence the lack of SNPs means that the element's missing. It doesn't mean that there aren't any SNPs there. This is the reference genome which was run through the procedure, so you can see there's no SNPs relative to itself, which is a reassuring finding. So removing all the messy accessory genes, we've inferred this tree, color-coded according to the origin continent. So the red ones here are the European ones. They sit basally in the tree and are actually relatively diverse. We have a green clade corresponding to South America and a blue clade corresponding to Asia. These are all the strains taken from the single hospital in Thailand. So immediately this implies that we have perhaps a European origin of this clone, because they're sitting basally in the most diverse in the tree. There are some discrepancies in terms of the geographical structure. We have this little group of Portuguese strains clustering with a South American. And this it turns out, these isolates corresponded to the second wave of 239 infection in the late 90s, early noughties in Portugal, which was caused by the so-called Brazilian clone on the basis of pulse field and resistance profiling. But this really nails down that this wave of infection in Portugal really is likely to have come from at least South America, if not Brazil. This is the reference isolate which caused the outbreak in London. Remember, it clusters with the Thai isolate, so at least that's we can say is of a Southeast Asian origin, that outbreak, although not necessarily Thailand, and more extensive data has actually pinpointed it to Singapore. This was a patient from an isolate taken from a patient in Denmark, and when we went back through the records, that's actually taken from a Thai person, so that was quite a nice moment. So by and large, it all sorts of seems to fit. There's a couple of things that we don't understand. There's Czech isolate here, but then, we're not going to explain everything. If we did, then no one would believe that it was real. So having got this tree, one of the interesting things we then did was to look at the homoplaces. There's very few homoplaces. It's a very, very well-supported tree. There's hardly any conflicts. So there's a nice number of homoplaces, 38, so that's enough to go and look at and see what they're doing. So homoplaces, if you're not aware, are mutations which have occurred independently on different branches of the tree. And that's either a result of coincidental mutation, which is a bit unlikely, or recombination, which is more likely, or perhaps they're adaptive changes, which, because they confer an advantage, are much more likely to be observed. So when we looked at the 38 homoplases, it turned out that 11 of them were already described in the literature as associated with drug resistance. So it really does seem to be a powerful way of from all this SNP data to zooming straight in on finding SNPs which are likely to be adaptive. Of course, there are other SNPs which are also homoplasic, which we don't know, which haven't been assigned a particular function. So, of course, in these data, in further data sets, it would be a very interesting approach to actually pull out the homoplases and see if there's any sort of reason why they should confer some adaptation. So this is the analysis which Nick as well described briefly yesterday in respect to his pneumococcal data. So once we've got the tree, then we can easily just plot the date of isolation of the isolate against the distance from the root. And the root, this tree was rooted by the ST8, which is the parental genotype, which acquired this big replacement, which I talked about at the beginning. So we can well root this tree. So it comes out, the emergence of this clone comes out actually sort of mid to late 60s, which kind of makes sense from what we know about the epidemiology of this clone, from when MRSA was first emerged, methicillin was first administered. And it gives us an estimated rate of mutation of about 3 × 10⁻⁶ per site per year, which is really fast. So we're looking at about 1 SNP every 6 weeks. If we look at the sample within the single hospital in Thailand, this is the Google image of this particular hospital in northern Thailand. If we look at the data, it splits nicely into two clades. And if we look to see where in the hospital those isolates were recovered, you can see that even though the numbers in this particular study of isolates were quite low, it does look promising that indeed there were differences within that correspond to different wards within the single hospital. So I'm moving on from this study now, and just quickly go on. So there's been another study which is not yet published, which has gone back to the same hospital, and there's 200 isolates from MRSA carriage, and the idea that they're concentrated on two ICUs, a surgical ICU and a pediatric ICU. And this is a tree of the SNPs, courtesy of Ben Cooper, who, and it shows very nicely that there's a difference between the blue ones here in the surgical ICU and the purple ones in the pediatric ICU. So you can see there's a few blue ones hanging out in the wrong ICU, as it were. And when we look actually at all the patient records and where the patients were staying in the ward and when the patients were staying on the ward, we can see that this difference is actually boils down to the arrival of one particular carrier, T126, as he or she is known, and who came in. So the different colours here, so this is time over the 3-month course of this study in the two ICUs. The different genotypes, the different colours represent different genotypes. The more similar the colour, the more similar the genotypes. So early off in the study, you can see that both ICUs actually had rather similar genotypes. It's purpley clade, and then this guy or girl, T126, came in carrying this bluey group, which is the other branch of the tree, and all the subsequent cases after, so this person stayed on the ICU for a long time and basically spread this blue genotype to all these other patients. So it's basically replaced the purpley genotype on this ICU with this different bluey genotype, which may have come from a different hospital, we don't know. So another study which I'm going to describe just quickly was aimed to fill this gap. We've talked about the global picture, and we talked about the picture within a single hospital. So it occurred to me that there's some wriggle room between these scales. So I set out to look on a nationwide scale, and then the country that settled on was Turkey. For a number of reasons, its position between Europe and Asia. It's been described in the literature as the epicenter of ST239, meaning there's basically all the hospital-acquired MRSA essentially is ST239 in this country. So the questions we wanted to ask is transmission on a national scale into and out of Turkey from the rest of the world, trying to look at relative rates of transmission between different hospitals and cities, and identifying epidemiologically relevant clusters both on local scales and on countrywide scales. So there were 4 hospitals, samples from four hospitals in this study, 2 from Istanbul on either side of the Bosphorus, 1 on the European side, 1 on the Asian side. There's another sample of 26 isolates from a hospital in Ankara, and a third sample of 50 isolates from a hospital over here in the West, Izmir. Oops. We also bulked up the tree with a few more global samples. And you see, first off, you can see very strikingly that the isolates from Turkey represent a very, very distinct clade in the global tree. The only ones that seem to be clustering with it are the ones from Eastern Europe. There's one from Romania I've highlighted here. But even other ones from the Middle East, like Syria, tend to sit on a completely different branch of the ST239 tree. So there's been very little. So all of the diversity within Turkey appears to be homegrown on a national scale. There's been very little import out of Turkey and very export out of Turkey and import into Turkey from the rest of the world. We can date the emergence of the major clades on the tree using BEAST, so we can nail down that this hypothesis that the thing emerged somewhere in Europe, and essentially there's been independent introductions from Europe to South America, to Asia and Turkey fairly early on in the evolution of this clade. So it's not like it's come from Europe into Turkey, into Asia. There's been multiple exports from Europe by the look of things. If we look at the Turkish isolates in a bit more detail, we can indeed identify, so these are colour-coded according to the hospitals on the map of Turkey down here. So we can indeed identify various clusters in each of the hospitals which appear to be epidemiologically linked. These ones were all in a cardio ward. These ones were all catheter associated. They're specific to a single hospital. But we also find a cluster which is common to 3 of the 4 hospitals down here. So we can identify clusters on local and national scales. And we can, of course, identify the SNPs, which are found in only these clusters. And as Tim said yesterday, oh my God, we can sit down with a glass of wine and work out what's going on there. Right, I'm going to zoom through these. So basically, the take-home message with this study, when we're thinking about hospital transmission, is that when we look at the 3 hospitals, 2 in Istanbul and 1 in Ankara, there's really, on overall, there's no difference between them. The difference between the hospitals is pretty much the same as the difference within each of the hospitals. So we have, on the whole, poor predictive power about which hospital a particular isolate will come from. If we throw in the hospital in Izmir into the pot, then this is clearly a much more distinct population, and you can see the map of Turkey here. The Istanbul and Ankara are joined by a really lovely road, so we'd expect more transfer between these 2 places than between Izmir, and that's what we find if we just look at basic FST statistics. The Izmir hospital is much more distinct. And that's it. That wasn't too bad. Thank you very much.